Okay, great. I will get going. So good morning, everyone, and welcome to the first CEBRA research webinar for 2025, or as they are known, CEBRAnars. So for those of you who don't know me, my name is Jessica May, and I am the Director of the Research and Innovation section in the Biosecurity Strategy and Reform Division. Before we get started, I wish to acknowledge the Ngunnawal people as traditional custodians of the land many of us are meeting on today, and recognise any other people or families with connection to the lands of the ACT and region. I wish to acknowledge and respect their continuing culture and the contribution they make to the life of this city and this region. I extend that recognition to the traditional custodians of all other lands on which our staff and participants are gathered today, and to all Aboriginal and Torres Strait Islander peoples attending today's webinar. I know a lot of you are tuning in from many different places and many different countries today. If you would like to acknowledge the traditional owners of those lands, feel free to pop it in the chat where you're joining from. So before we begin, can I please ask everyone to ensure your microphones are muted and your cameras are off? This CEBRAnar is being recorded for those who can't make it today. There will be an opportunity at the end to ask questions after the presentation. So you can either use the hand up gesture or post a comment in the chat on the side as we're going, and then I will come back to them at the end of the presentation. For newcomers, the Centre of Excellence of Biosecurity Risk Analysis is a long-standing biosecurity research initiative. It's currently hosted by the University of Melbourne. CEBRA plays a vital role in providing the Australian and New Zealand governments with expert biosecurity risk analysis and advice that helps inform a range of biosecurity risk management activities. So today we're in for a treat as we are joined by Associate Professor Richard Bradhurst, who will be presenting on the development of a decision support tool for potential outbreaks of lumpy skin disease in Australia. So Dr. Bradhurst is Chief Investigator at CEBRA and specialises in the fusion of multiple modelling approaches to simulate the spread and control of emergency animal disease, plant and environmental pests and human disease. Richard is the co-creator and principal developer of the Australian Animal Disease Spread System, the European Transboundary Animal Disease and the Australian Plant Pest and Disease Modelling Frameworks. Richard was also the recipient of the Dr. Kim Rittman Award for the Science and Innovation at the 2022 Australian Biosecurity Awards. So without further ado, I will hand over to Richard to start the presentation.
Thank you so much, Jessica. And just get organised first. Now, how did I do? Hopefully the right screen is appearing there.
Yes, yeah, all see the presentation.
All good. Yeah. Excellent, excellent. I've got so many screens here. So look, it's a pleasure to be here, and thank you so much to everyone that's tuned in. I really appreciate these opportunities to share work that's gone on behind the scenes, and it's certainly not just me. So, oh, now, is that clicked? What screen are you, what's page are you seeing now?
Front page, the front page, title page.
For some reason that's not progressing. Excuse me for a moment. I hope that's better. Yeah, I just had to restart PowerPoint. So yes, it's a project group. It's not obviously not just me muttering in a dark room. So just a shout out to first of all, the DAFF for funding this project to develop a decision support tool for lumpy skin disease outbreaks in Australia. And I'll come back to that at the end in the acknowledgements, but just to have that up front. So a quick primer on lumpy skin disease. It's a pox virus that affects domestic and wild bovines. So in the context of Australia, That would imply our cattle, domestic cattle, potentially feral cattle, and feral Asian water buffalo. So after an animal's been exposed to the virus, there's an incubation period of 4 to 14 days. So an average perhaps 7 days, at which point fever will arise. There'll be discharge from ocular, oral, and nasal discharge. And then the most profound clinical signs is, of course, the lumps or the nodular lesions. These can be 2 to 5 centimetres in diameter. They can progress to necrosis. They can track secondary bacterial infection. So the animal does poorly when they're exhibiting clinical signs. There'll be depression, loss of condition, and a reduction in milk yield. Mortality rate is usually low, so 2 to 10%, but a lot of these type of parameters do vary depending if you're in the field or in an experimental situation. Once an animal is infectious, it'll be so for 9 to 23 days, and lumpy skin disease will transmit nose to nose, so it's through direct contacts as well as indirect contacts. But the prime driver for spread is mechanical transfer. So insect vectors, they don't become infected themselves, they don't replicate the virus, but they transfer the virus mechanically from an infectious animal to a susceptible animal. And the sources of that infection will be the lesions and particularly the scabs for blood-feeding insects. The blood is a rich source of virus, and of course the secretions. Now, if you've not heard of or visited the FAO Empress website, it's rather nice. I went there just the other day to prepare for this talk and did a query on lumpy skin disease outbreaks. So as of 2019, there were reported outbreaks in Bangladesh and China. There was northern India as well, but I didn't include that in my map, so that's just off the screen. So at that point, just the 3 countries in Southern Asia. I'm not looking at Europe at this point. End of 2020, that had expanded to Nepal, the Kinmen area of Taiwan, which is just off the mainland China, Bhutan, Vietnam, Hong Kong, and Myanmar. End of 21, explosive growth in Thailand, Cambodia, and Malaysia and Laos, as well as cases reported in Pakistan and Sri Lanka. By the end of 22, it had gone to Singapore and Indonesia. 23, there were more reported cases in Indonesia, as well as 2024. So this is this looks diabolical, but this is the cumulative reported cases. So it's not the necessarily the active cases or the new cases, but this is the cumulative over time. And I've got a little graphic there that I made just to show the progression in cumulative cases over that 5-year period. So as you can see, creeping ever closer to Australia, which is a cause for concern because lumpy skin disease is a notifiable disease. It causes a great impact to a country such as Australia. There'll be obviously the production losses, the cost of control, but importantly the trade impact, and that we would lose our lumpy skin disease free status. So the question on the table is, what might an outbreak of lumpy skin disease look like in Australia? And it's not a straightforward question. There's lots of variables influencing that the potential pattern. So there'll be what are the insect vectors in Australia that might be competent? What's their distribution and abundance? Where is the cattle in Australia? Farming densities, different production systems will have different levels of exposure to insects. The Bos taurus is more prone to clinical disease than the Indicus. And we've got quite different livestock movement patterns across Australia. So it's a very diverse country in the sense of farming patterns and marketing systems. Feral cattle may play a role, water buffalo and spread. We're not quite sure about that. And then once control programs underway, variables include how soon will we notice it, where will we notice it, what options are on the table for response, and what resources might be required to enact that response. So overall, it's a complex problem, time and space. There's a lot of uncertainties and lots of regional and seasonal heterogeneities. So luckily, we're not starting from a blank page. So thanks to the foresight of the Australian Department of Agriculture, Fisheries and Forestry, in 2012, they funded a development program to build a decision support tool for Australia. What came out of that project is the ADIS model, Australian Animal Disease Spread Model. And I always like to shout out that that core group back in the day of myself from University of New England, Graham Garner, Sharon Roche, Ian East, and Rachel Iglesias. So together, we worked very closely and came up with the Australian Animal Disease Spread Model. And I never hold back from an opportunity to preach a righteous wind up about the ADIS model. So if you have not seen it, this is the ASF model. It's obviously highly visual. And one thing you might notice is that there's not a real pattern forming, like these blotches of color are different every time. And that's because it's a stochastic model. So the model explicitly honors uncertainties, heterogeneities, the wibbly-wobbly aspects of life. When we run out of resources to enact a control program, or we get a clinical report of disease that's a red herring. So these things that get in the way of a response, we honour all that, and we get different outbreaks every time we run the model. So here is a visualisation of one particular iteration, and it shows you the infection network, the different colours on the screen, some of the control designations that get assigned to properties during a response. So certain colours will be that a tracing activity has identified an interesting premise that may have had infection moved onto it or moved off it. There might be clinical reports, movement restrictions. So this is a snapshot of that particular iteration, one of many. This shows you the unknown versus the known. So the orange is the known outbreak, which is only known to the model. And the red is the known outbreak, so the reported declared infection. So that's the battle between the good and evil, or the unknown and the known. That's an epi curve showing us the impetus of the outbreak, so new cases per day. And these displays here are quite interesting. They give us an insight into how well we're doing with respect to our resource pools. So in that particular graphic, those little blotches of red, that's telling me that for this particular outbreak of ASF in Queensland, at this particular day that I've frozen it, we're short of surveillance teams. So the red bar shows that we've gone in deficit, and there's jobs that need to get enacted. So there's reports of clinical disease, but we don't have a surveillance team available to go and have a look at that on that day. So that feeds very much into how well we do in a response. So this resourcing is a critical aspect of the ADIS model. And we keep track of the cost of every single action that's simulated. So we can build up over time an idea of the potential cost of control. Now, as I mentioned before, it's a stochastic model. So you've got to, for any given scenario, in this case, it's a small commercial piggery in southern Queensland. We rerun it over and over again. And we build up distributions of outcomes. So what you see forming there in the blue histogram is the range of outbreak durations. So right now, we've done about 85 iterations. Our median duration is 83 days, I think. And the red histogram is the number of IPs, so the median there is 16 or 17. So why did we go to all that trouble of building that model? Because complex simulation models are difficult to build. They take a while. But the benefit is that once you've built it, you put a lot of power back in the user of the model. So for every question that a user might have, we don't need to reformulate the model. We don't need to redo the maths and the stats in computer science. We'd like a flight simulator. We just configure the problem that we're looking at. So we can ask, the user can ask these questions directly to the model. Now, since that initial FMD decision support tool development, we've looked at other diseases in Australia. So we've looked at blue tongue, so biological vector-borne disease. We've looked at the potential role of feral pigs, both at FMD and African swine fever. The project that I'm sharing with you today, lumpy skin disease, and we've started looking at JE, that's the current project. New Zealand, Mary Van Andel put together an FMD model for her PhD project, and CEBRA looked at Mycoplasma bovis. So we built a model to help them, to assist them in their background surveillance program running to see if there is any residual evidence of Mycoplasma bovis in their cattle population. Blue tongue in the USA. In Canada, they've renamed the model Candace, which I think is rather nice. So they're developing or configuring an FMD model as we speak. In Europe, they badged it as EOFMDIS, and diseases that have been looked into there, FMD, classical swine fever, both in domestic and animals and wild boar, sheep and goat pox, and PPR. At the moment, Professor Mark Stevenson from the vet schools building a rabies model for Indonesia. And the model's also been expanded to invasive species. So we've looked at some invasive ants, fruit fly, some grain pests, and there's also a human variant of the model. Now, these are all the one model, and that's by design. So when a user receives the ADIS model, the model is put in a mode. So it's either contagious livestock disease, vector-borne, wildlife spillover, plant pest, human disease. And you configure the model with a database, four config files, text files, and you end up with one of the coloured boxes on the screen there. So this is very handy, having a single model doing all these different things, because it means that the philosophy of ADIS is that if any one country improves the model with functionality, it feeds back into the core, to the Java code, and all the users in the tribe have access to that new functionality. So the functionality is in the radioactive core at the bottom, and the data, the configuration, and modelling outcomes are in the top private boxes in the separate countries and the separate projects. This is a set of collaborators, and I think there's about 25 countries that are using the ADIS or ADIS derivatives at the moment. So at last, we get to the topic. And so our ADIS development project ran from 22 to 24. And the first step is what are the vectors that might contribute to lumpy skin disease? So this was some great work done by Emily Sellens of DAFF and James Kamak of CEBRA. So it was like a little sub-project. So they went away, did some great work doing literature reviews, consultation with our Australian entomologists, and looked into things such as habitat suitability of candidate vector species, what occurrence records are available in Australia, what's the known relationships between certain species with cattle, and biting and feeding behaviour. And the goal was to come up with 12 exemplar vector species. So this, we're not saying that these are the only species that could contribute to an outbreak, but the consensus was that these are potentially 12 important species. We know that they're present in Australia. We know a bit about their distribution and abundance, about their behaviour, their ecology. We know about their relationships with cattle, their biting behaviour. And so we identified, or Emily and James and experts identified these 12 species that we would pursue in the project. So 4 fly species, 4 midges, and 4 mosquitoes. Now, luckily with insects, we can approximate habitat suitability by their response to temperature. So it's feel like a slightly simpler problem to consider than a vertebrate species. So there was an activity undertaken to come up with some habitat suitability models in the context of a national grid 4 by 4 kilometre cells, and we were blending climate data and occurrence data, and the result of that is 12 sets of monthly maps, and we've got this new concept called pressure. So pressure is a combination of a vector's estimated distribution, abundance, and activity. And again, we can proxy activity through temperature response curves. So species will have a temperature range that they do best in, and outside those ranges where they don't do so well. So quite simply, we've ended up with a score between 0 and 1 for each grid cell in Australia, pertaining to the likely pressure that particular species might influence on that month in Australia. Now, the next step was to aggregate our exemplar vectors into a single, what we referred to as the Franken vector. So an imaginary aggregative vector that combines the pressures that these 12 individual exemplar vectors might exert. And again, Emily and James went to the experts and to determine the relative importance of the vectors. And so I won't go into too much detail, but loosely, it was buffalo fly more important than the midges, more important than the mosquitoes, then the stable fly, the bush fly, and the house fly. And the sort of criteria that they looked at was feeding preferences, so known associations with cattle, biting rates. And so there's an interesting one, the likelihood of interrupted feeding. So if a species has got a sharp, painful bite, for example, they might be interrupted midway through a blood meal, and then they might fly off to another host to continue the meal. So there's potential there for transfer of virus. Also, the typical ratio of vectors to host, and how long the virus might remain active on a particular vector. So that aggregation process, we ended up with monthly national vector pressure maps, and that's a display on the right there, just cycling through the months in Australia. So it's the darker colours is the low pressure, and as we go up through brown, orange to yellow, we get the higher pressure. So you can see in the tropical areas of Australia, we pretty much have year-round high vector pressure. But a lot of variability in central and southern Australia, where we get seasonal variations in the vector pressure. And so one final call out to Emily and James for such great work in that sub-project. So the next problem to solve is where are the domestic cattle? So, luckily we've had a bit to do with representing cattle in Australia in our FMD model. We've in ADIS with our epidemiological unit of interest is a herd. So we don't look at individual animals per se, but we look at them in the context of a herd. So we say a herd is a group of commingling animals, same species, same production system. Examples in the LSD model, dairy herds, feedlot, intensive beef, extensive beef, and smallholder. So on the right there is a density map of cattle in Australia, but in the modelling context, it's an individual-based model. So every single herd is represented individually as a point on the map, which you can interact with. Each herd's got its own story of how big it is, where it is, the production system. And each herd has its potential to acquire infection. So don't worry too much about the details there. Let's just focus on the picture. So we represent the potential spread of lumpy skin disease virus within a herd through a very standard tool called a compartmental model. And quite simply, what that is we view a herd as the members of the herd are going to exist in one of those boxes at any given point. So the S box is susceptible. So at the start of the model's run, all the animals get loaded into the S box. If they get exposed, challenged by a virus, so for example, that evil vector cloud that you see there might convey some infectious material. They become exposed to a virus, so they transition to the E box. Once their latent period has expired, they'll transition to the I box for infectious, which means they're shedding virus and they're posing a risk to other animals. You'll see there's a connection between the I box and the S box through the vector cloud. So that conveys this idea that predominantly we're going to be influenced by the level of vector pressure with that will influence the vigor that the virus will go from the eyes and influence the susceptibles. At the end of the infectious period, the animals will be either recover with some natural immunity, so they'll be in the R box, or they'll be deceased and go into the D box. So it's a very simple concept. It's implemented with differential equations, which you don't need to worry about, but they govern the flow of the animals through those different boxes over time. So that's part of the configuration or the parameterization of that within herd submodel. And if we solve those equations, we get these lovely curves, which just simply is a mathematical projection of what might happen inside that one herd if lumpy skin disease is introduced. So the different colours represent the different types of prevalence over time. So infectious prevalence or clinical prevalence. And there's other prevalences there too, but I won't go into that. So the next step is how might an infectious herd influence a susceptible herd? And ADAS offers a bunch of different pathways to simulate that, but in our particular application today, we're just looking at movement of infected animals, potential for amplification with those animals being batched and redistributed out of cilia, and insect vectors. So again, you don't have to worry too much about the maths here. Let's just focus on the English. So if we want to model the short-range spread of lumpy skin disease between two separate herds, the first thing we're going to want to know is the infectiousness of the source herd. So that's provided to us by those little coloured boxes we just looked at. So that's giving us a dynamic, day-by-day projection of the level of virus present in the herd. The local vector pressure, which is courtesy of the vector cloud that we've also looked at. So that will influence the likelihood of virus jumping between two herds. And then finally, a space or a distance term. So, and very simply, we say that the further away that a susceptible herd is from an infectious herd, then the probability of transmission decreases. So it's a nice simple concept, and it's called a kernel. And it's a very standard way of representing transmission. Sometimes when we're not quite sure as to the exact nature of the transmission, we can just parameterize a very general kernel because we know a rate of spread, for example. Now, because we don't have data in Australia, it's calibrated through overseas experience and local expert opinion. So if we were to get lumpy skin disease, it'd be quite straightforward to recalibrate this based on outbreak data as we learn more about the behaviour of an outbreak in Australia. Now, modelling longer range spread of lumpy skin disease, here it's a little easier to get your head around because we have got good data on where cattle move in the country. So it's quite plausible, given we've got an incubation period of 4 to 14 days, that animals could be infected but not yet showing clinical signs. They could be loaded on a truck. We do have quite long distances in parts of Australia for cattle movements. At the end of the journey, they get unloaded and potentially encounter vectors at the destination that could interact with the cattle and continue the transmission process. Now, we know with other models, we've done a fair bit of work, but with this project, we improved it. So we looked at export facilities. So we model explicitly farm-to-farm movements, farm-to-abattoir, farm-to-export facilities, and farm-to-sale yards, and then on subsequently to farms, abattoirs, or export facilities. And the interesting thing here is that once you've moved the animals to each of those premises, then they might have a new story to tell, depending on what vector pressure is being exerted at those destinations. Now, this is really the heart of what we do, because modeling the spread of an outbreak, there's a lot of uncertainty, there's sometimes heroic assumptions, there's heterogeneities, and that sounds bad, but it's not, because the goal here is just to model plausible spread of a pathogen in a naive population. No one's going to know where a disease is jumping to next week or next month, but we can simulate a range of possibilities. So that's the beauty of a stochastic approach, an individual-based approach. We can get a smorgasbord of possible outbreaks, and we get an idea of the most likely outbreak, the least likely. And it's against that sea of uncertainty of the possible outbreaks that we apply and test our response options. So the heart of what we do is being able to do experiments on things like different options in movement restrictions, different sizes of zones, the duration of a zone, tracing effectiveness, surveillance protocols, the sort of stuff that we want to get a handle on how well we might do. We can test these things against a range of different outbreaks. Now, with this project, we added some new stuff. So we've got vector control in for the first time as an option for experimenting with. We've got a suite of options to do experimentation on possible vaccination options in an outbreak. And importantly, we got a better handle on the north. So because we're focusing on on the perhaps likely entry point for disease from the north, we've got a better handle on wet-dry influences on mustering effectiveness, and then how that might influence the ability to carry out surveillance and measures such as destruction and vaccination. So just to show that I'm not fibbing, here is a movie of the model that we developed. And in this particular case, I've dropped lumpy skin disease virus in an extensive cattle property northeast of Tennant Creek. And we can see the red arrows, that's where movements have occurred with infectious animals. So one shot down to the Wyandra export facility, one shot up to Connemara sale yard, and one shot up to Livingston export facility up near Darwin. And I'm just replaying one iteration just so I can talk about it. That's why it's not changing here. But what's interesting is that we've got these two mechanisms being illustrated in the movie. So and I'll start again, when we see the orange, that's the vector-borne spread locally. So we can see that at the Tennant Creek site and down at the Wyandra site. And then the control is exerted, and in this case, we control the outbreak. So again, that's the infection network that's generated for this particular iteration. The fight of the unknown versus the known. There's a new curve for this model, so that shows us the vector pressure over time in that area. And this is an interesting part. So this was after 94 of 500 iterations, and we're getting more variability. So you remember with the ASF movie that I played, we were getting quite a nice normal distribution after 100 outbreaks. But here we're seeing much more of a long tail forming. So that's informative, instructive. Now, the tool's been built, and so the outcomes that arose from the project is a new decision support tool. So just one of many tools that's available for decision makers, stakeholders to better prepare for LSD outbreaks. It allows a user to experiment with a broad range of incursion scenarios. So you think that all the possible herds, we've got 65,000 odd farms in the model that have cattle. Theoretically, any one of them could experience infection of lumpy skin disease on any given day of the year. So there's millions of different permutations. So the model allows us to do these sweeps. You know, we can pick a region, we can scatter lumpy skin disease in certain types of farms, different times of the year, and very hard to get a handle on a complex time-space problem without that ability to allow the regional and seasonal variations to be taken into account. The business end of things is really for the experimentation with our options of response, so size and duration of restricted areas. I didn't mention that we have naughty dial in the ADIS model, so we can change our confidence level in how well movement restrictions will be obeyed. So if you like, the porous nature of a quarantine, in that there may be illegal movements out of a restricted area, or there may not. So we can dial up these illegal movements and see what the consequences are. We can dial up the reporting of red herrings. So what happens if we just get loads of reports of clinical signs that aren't lumpy skin disease, and we've wasted a surveillance visit? Importantly, we can do experimentation on vaccination options, when we might trigger vaccination, the type of vaccination, the herd types to focus on, what type of vaccine bank might be required. We can try different vaccine effectivenesses, vector control options, what might trigger it, how effective they might be, resourcing requirements. So all of these are inputs that they go into the concurrent nature of an outbreak. So all of these need to actually be viewed altogether in a simulation to allow them to interact and to fight. And what comes out the other end is the emergent behaviour of potential outbreaks. New methodologies that arose. So it's the first time we've looked at a mechanical vector. We've looked at biological vectors with blue tongue. So we've got some new tools in our national toolbox for representing mechanical vectors, and methodology for deriving pressure maps, which is very nice. Methodologies for transmitting disease through mechanical vectors. And we've got we think a better representation of northern cattle herds now. So we actually view northern herds as comprising of many mobs, and each mob can have its own infection story. So you can have mob-to-mob transmission in northern cattle herds, as well as farm-to-farm. An important one is mustering limitations. It's very bad to make the assumption that we'll always have perfect access to stock in the far north, in the tropics. So we allow these imperfections in surveillance, access to stock to be seasonally adjusted. And I do want to mention that what I presented today is not just for Australia. So the investments that DAFF make are international scope. So as I mentioned before, the new functionality goes into the ADIS core, and it's automatically available to other countries. So, for example, Denmark is about to launch into the construction of a lumpy skeesden model. They inherit the methodology, the functionality, because the EFMDIS is an instantiation of the ADIS core. So this is a tremendous model, I think, of international sharing. So enabling us to solve, not keep resolving the same problems over and over again. And drawing to an end. So once again, just a shout out to the project team. So this was a great project to be involved with. So in CEBRA, James Kamak, DAFF, Sharon Roche, Emily Sellens, Hatham Taha, and Andrew Breed, a consultant, is the ever young Graham Garner. And we got some great engagement from WA through Jamie Finkelstein and Graham Macra. And we've really valued the contributors or contributions from other government animal health staff, so NT, Queensland, New South Wales and Victoria. CSIRO, Debbie Eagles, Kelly Stanger and Tim Bowden. Expert entomologists, Glenn Bellis, Peter James, Greg Devine. Pirbright, Pip Beard and Henry Mandioki. And DAFF, Jared Sanderson. So that's that. I think that's the end of the presentation, and happy to take some questions.
Fantastic. Thank you so much. That was wonderful. Just a reminder to put any questions in the chat down the side and I will read them out for Richard to answer, or otherwise raise your hand and I can move to you directly. But I will kick off. I had a question. So what's the plan for next? What do you want to do next?
Yeah, so I'm really just sharing the development of the tool and the assumptions, and of course these are up for challenges by people. Like if you saw something that worries you, then that we welcome feedback. So this was really just to announce that the tool is developed, and the next stage is to really use the tool in anger. So DAFF, we've run some scenarios for DAFF, and DAFF will be processing those results, and that will, at their choosing of the timing, that'll be made public. But on a personal note, there's some gaps that we haven't addressed. So we haven't actually incorporated the potential role of feral cattle and Asian water buffalo. So that would be, I think, an interesting and worthwhile thing to include. And I think we've got a reasonable handle on northern distribution of water buffalo from memory. So that would be cool. And we'd look more into vaccination options and vector control and just get a better handle of what's on the table, what tools are available if we do get lumpy skin disease.
Fantastic. So I'll hand over to Rob Williams, who has their hand up to ask a question.
Yeah, thank you very much, and really appreciate that presentation, Richard. It's excellent. That model looks fantastic. We have a significant task under the National LSD Action Plan to develop a vaccination strategy. AusVet Plan itself has a strategy, but it's probably not as prescriptive or as detailed as people would like. The group that I was chairing for a little while, that I'm still going, but sort of towards the tail end, was tasked with that, but we struggled in the strategic space without any sort of inputs. I was just, the simple question I have is, do you have any recommendations for how to use this model? I mean, you obviously articulated what it can do, but as part of the process of developing a more detailed strategy that give people some comfort that in particular scenarios, this is how we would employ vaccination.
Yeah, that's certainly on the table. I know DAFF are very, very interested in that. And this is where the model can help because of the regional and seasonal heterogeneity. So, as an example in the FMD space, our modelling has shown clear suggestions that under certain circumstances in FMD, vaccination can help. In other circumstances, it may not help at all. And I'm not saying that will be the case with lumpy skin disease, but it's just this idea that you can't just have a blanket statement of this is what we will do. If a disease gets in, it's really going to matter on where it gets in, the production systems as to what options are being used. So I think part of where we will go is doing, broad sweeps of simulating outbreaks across Australia and just getting a read on how the outbreaks are differing. And, it's not for me to in any way comment on policy, but just to provide a tool that can be used to help in that area.
Thank you.
Okay, great. We have another question from Peter. So he was interested in your approach about combined and temporarily variable. or temporarily variable vector pressure? Will you be publishing these methods somehow so the process could be emulated for other vector species?
Yes, so that's a great little component that we can shelve off and publish without any privacy concerns or sensitivities, because it is just really a ecological and modelling component. So very much that it will be available as part of this sharing paradigm, and that would, Emily and James would lead that, I think, to produce a paper that just goes through the methodology, and then eventually that would be part of the greater set of learnings. But I think there'd be value in doing that first. And I think Emily has presented that in isolation at Science Week, so she went through some in more detail than I'm capable of that work. So I'll definitely encourage Emily and James to pursue that in a standalone paper.
Fantastic. Does anyone else have any other questions that want to raise a hand? I was kind of a bit more interested in, you were talking about adding in, so feral cattle, and you had a fair bit of data there about their movement and things. Whereabouts are you getting that from? So,
oh well, actually, it's feral pigs have been our main focus.
Yeah.
So
Yeah.
I don't personally have information on feral cattle. I've just heard anecdotally that there is data available on water buffalo, and I've seen distribution maps. So if I can draw the analogy of what we did with feral pigs, it's a standalone process to get a handle on distribution and abundance and activity in time and space. And then the model does have different ways of looking at the world. So we can look at just a wild population in isolation and drop diseases in that and replicate group to group transmission. For example, sounder to sounder feral pigs. But I guess we'd have to sort of investigate how the feral cattle hang out. If they hang out in mobs, if we can characterise them as standalone, homogeneously mixed groups, like we do with sounders and farms. So that if that opportunity arises, that would be an interesting, I think, worthwhile pursuit, and that the methodology again would be available for other interested parties to share in that.
Yeah, I was just thinking there'd be a lot of transferable information that you could reuse if we were to map that. It's a really interesting sort of area.
Yeah, and as I mentioned, that we want to tie this in with Biosecurity Commons. So, where there's no sensitivities and we've just added value to data sets, we very much want those to be made available and not always have to be re-engineered. So that the natural home for that is the Biosecurity Commons space. And so that would be our tactic there.
Fantastic. We just have another question from Thomas asking if there's any coming ADIS project addressing aquatic animal diseases.
There's not. There's been rumblings from time to time. So the short answer is no, but I wouldn't rule it out. If there was something we could do with a straight face that was useful, like if we could vulgarly simplify three-dimensional water space in the model and do it efficiently and add value to a space, then yes, we could hopefully help, but there's no plans at the moment for aquatic disease.
Okay, great. I'll do one last call out if anyone wants to put their hand up. Otherwise, we will move to wrap up. So, what is it? It's 7 seconds of uncomfortable silence. No, I think that's it for everyone. You must have answered all the questions in your presentation, which means it was very thorough. So thank you so much, Richard. Really appreciate your time today. It's been fantastic. So